everyone, we are Jessica and Raquel from the Train2Target network. We are going to talk about the RCS system in bacteria. Bacteria are very small organisms. They can be found everywhere. As any other microorganism, they need to protect themselves. That's why they have a cell envelope. There are two types of bacteria, depending on the physiology of the cell envelope. The gram-positive bacteria have a cell membrane and a thick layer of peptidoglycan where we can find proteins, teichoic and lipoteichoic acids. The gram-negative bacteria have a cell membrane but also a second membrane called the outer membrane. Both membranes are separated by the periplasm where there is a thin layer of peptidoglycan. In the outer membrane, we find lipopolysaccharide and surface-exposed protein. Today, we will focus on the gram-negative bacteria. As an example, we will talk about the Enterobacteriaceae family, such as Escherichia coli. In the inner membrane, we find integral proteins. Beta barrels can be found in the outer membrane. Soluble proteins are in the periplasm. As for lipoprotein, they can be attached to the inner membrane or to the outer membrane facing the periplasm. Bacteria use different systems to defend themselves. The regulator of capsule synthesis, or the RCS system, is one of them. It is conserved in the Enterobacteriaceae family. It's a two-component system formed by an outer membrane sensor called RCSF. RCSF will sense the defect in the peptidoglycan and in the outer membrane. Therefore, it will interact with IgA. This interaction will activate the response, so the RCSC will be autophosphorylated, and the phosphate group will be transported by the RCSD to RCSB. RCSB will be activated as a homodimer or heterodimer with RCSA. These two forms will activate the transcription of many genes that are involved in the biofilm formation, the periplasm quality control, the homeostasis, the surface antigen production, and the motility of the bacteria.
Now, let's see a bit more in detail how this outer membrane lipoprotein, RCSF, senses envelope damage by monitoring the activity of the beta barrel assembly machinery BAM. In the image at the left side, we can see what happens in the cell under normal conditions. RCSF will interact with the central component of the BAM machinery, BAM-A, and BAM-A will funnel RCSF into outer membrane proteins like OMP-C or OMP-F. In this complex with OMPs, RCSF will be partially surface exposed and it will be occluded from IgA, the repressor of the RCS system. Therefore, under non-stress conditions, the RCS phosphorylase remains shut down. Now, let's take a look at the image on the right. Envelope damage can be caused by many events, including some antibiotic treatments. When the cell suffers from an increase in the envelope stress, the newly synthesized RCSF can no longer bind to BAM-A and therefore remains free in the periplasm where it is able to interact with the periplasmic domain of IgA. This interaction between RCSF and IgA triggers the activation of the RCS phosphorylase and therefore the cell envelope damage adaptation of the cell. Understanding how the cells defend themselves against an antibiotic treatment will help in the development of new drugs to fight antibiotic multi-resistant bacteria.